Good afternoon, students. Further, we will study the types of embryo sac in angiosperm. In majority of the angiosperm, the embryo sac develop from only one of the four megaspore formed by the meiotic division of the megaspore mother cell. The formation of female gametophyte from two or all the four megaspore have been observed in many species. Embryo sac have been classified into monosporic, bisporic or tetrasporic type depending upon whether 1, 2 or 4 megaspores are involved in the formation of an embryo sac. The different type of embryo sac are as they are. The first is the monosporic embryo sac. This type of embryo sac is derived from one of the four megaspore. It is characterized by genetically identical nuclei as it is derived from through mitosis of the single nucleus. Monosporic embryo sac are of two types. They are polygonum type. It is the most common type and was first observed in Polygonum. The embryo sac developed from a single chalazial megaspore and is 8 nucleated, comprising 3 antipodal and a binucleated central cell and a 3 cell egg apparatus. Second one is Oinothera type. In this type, the embryo sac developed from a single micropolar megaspore and is of 4 nucleated, organized into 3 cell egg apparatus and an uninucleated central cell, that is 3 plus 1 arrangement. This type of embryo sac is found among the member of Onagraceae. Second type is bisporic embryo sac. In the plant bearing bisporic embryo sac, the megaspore mother cell undergoes first meiotic division, which is followed by wall formation, giving rise to haploid dyad cell. One of the dyad cell is the functional cell, whereas the other dyad cell facing the micropilar end degenerates. The functional cell divides by second meiotic division, which is not followed by wall formation. Hence, the binucleated functional megaspore is formed. Both megaspore nucleus participate in the embryo sac formation and divide by 2 mitotic division, forming 8 nuclei. The 8 nuclei are organized into 3 cell egg apparatus, a binucleated central cell and a 3-celled antipodal. A bisporic embryo sac is constituted of 2 genetically different nuclei. Hence, of the 8 nuclei formed, 4 are of one type and the other 4 of different type. Bisporic embryo sac are of two types. They are of allium type. This type of embryo sac was first observed in Allium fistulosum by Strasburger in 1879 and is found in all the species of Allium. The embryo sac developed from the chalazial dyad cells. Distribution of the nuclei followed the general pattern, that is 3 plus 2 plus 2 arrangement. Endymion type. This type of embryo sac developed from the micropilar dyad cell. In Cincandra chinensis, the derivation from the normal bisporic has been observed and the mature embryo sac is bisporic but 4 nucleated. Tetrasporic embryo sac. In such case, the megaspore mother cell divide by meiotic division and neither of the two division is followed by wall formation. So, the 4 haploid nuclei remain in a common cytoplasm. Such a 4 nucleated embryo sac is known as cynomegaspore. All the 4 nuclei of the cynomegaspore participate in the formation of embryo sac. Hence, a tetrasporic embryo sac is a more heterogeneous due to the presence of genetically different nature of the nuclei. Tetrasporic embryo sac was further classified depending upon whether the nucleus fusion occur between the nuclei, the number of post-meiotic mitosis in the cynomegaspore, and final the organization of the embryo sac. First is the nuclear fusion does not take place. They are further divided into Adoxa type, Plumbago type, Pinia type, Drusa type, and Pepermonia type. Nuclear fusion does not take place. In the Adoxa type, the four nuclei undergo a single post-meiotic mitotic formation of 8 nuclear embryo sac. The 8 nuclei are organized into 3 cell egg apparatus, 2 nucleated central cell and 3 cell antipodal, that is 3 plus 2 plus 3 arrangement. It was reported by Johnson in 1818 in Adoxa moschete linam. Second is Plumbago type. The four nuclei undergo only one post-meiotic mitosis resulting in 8 nuclei. However, the 8 nuclei are distributed in the form of an uninucleated egg cell, a 4 nucleated central cell, and 3 nuclei are cut off as a peripheral cell, that is 1 plus 4 plus 3 arrangement. In Pinia type, in this type, the four nuclei undergoes two pre-meiotic mitosis resulting in 16 nuclei which are arranged in an unusual manner. The mature embryo sac comprises a 3-cell egg apparatus at the micropylar end, 3-cell group at the chalazal end, 2 lateral group of the 3-cell each at the central group of 4-cell functioning as a polar 3 plus 3 plus 3 plus 3 plus 4 arrangement. Stephen in 1900 described this type of embryo sac in the 3 genera of the family Piniaceae, which Pinia, Brachysiphon and Sarcocola. Fourth one is Drusa type. In Drusa type of embryo sac development, post-meiotic mitosis are similar to Pinia type, resulting in 16 nuclei which are arranged as a 3-cell egg apparatus, 2 polar nuclei and 11-cell antipodal, that is 3 plus 2 plus 11 arrangement. In Pepermonia type, this type of embryo sac is similar to Pinia type. That is, it consists of 16 nuclei which are arranged in the form of a 2-cell egg apparatus, 6 peripheral cell and a central cell having 8 polar nuclei, that is 2 plus 6 plus 8 arrangement. Thank you.